Hello everyone, today we are going to learn about cell wall. Plant cells are surrounded by an extracellular matrix known as the cell wall. Cell wall is the outermost strong layer of the plant cell. It is present outside the plasma membrane. It is a unique feature of plant cells which is absent in animal cells. It is important for regulating cell volume and determining cell strength. It is also important for giving tensile strength and limiting plasticity to the plant cells. So, in this module we are going to learn about importance of plant cell wall. Learning objectives of these modules are introduction of cell wall, primary cell wall, hypothetical model of cell wall and composition of cell wall. Introduction. Plant cells have a unique feature unlike animal cells, that is presence of cell wall. All plant cells are surrounded by an extracellular matrix known as the cell wall. Cell wall plays important role in plant growth, development and defense responses. Cell wall plays important role in regulating cell volume and determining cell shape. Cell wall is responsible for tensile strength and limited plasticity of the cell. Plant cell wall is essential for many processes in plant growth, development and reproduction. It acts as a diffusion barrier for large size molecules and as a structural barrier to pathogen invasion. Electron microscopic observation and use of different strains indicate a non-uniform varied nature of the cell wall in different cell types. Like epidermal cells, there are two types of cell walls, primary cell walls and secondary cell wall. Primary cell walls deposited during active growth and relatively unspecialized, whereas secondary cell wall formed after the cell growth during differentiation. Cell wall is permeated by small membrane lined channels called as plasmodesmata. Primary cell wall. It is the first wall laid down at the end of cell division, many times referred as phragmoplast. It surrounds the growing cell in the young tissue, meristematic cell and present in all types of tissues. It is composed of 90% carbohydrate and 10% proteins. Organization of primary cell wall. Three classes of polysaccharides make up the primary walls, namely cellulose, hemicellulose and pectins. The primary cell wall can be divided into two types, type 1 primary cell wall and type 2 primary cell wall. Two main components of primary cell wall are cellulose and matrix. Matrix is formed by hemicellulose, pectins and proteins. Basic structure of primary cell wall shows presence of cellulose microfibrils embedded in highly hydrated matrix. Matrix consists of hemicellulose, pectins and small amount of structural proteins. Matrix is highly hydrated gel phase in which cellulose microfibrils are embedded. Primary cell wall can be divided into types based on their composition. Type 1 primary cell wall present in all flowering plants except the grass family. It shows presence of cellulose, hemicellulose, that is xyloglucan, pectin approximately 22 to 35%, homogalacturonane, HGA, rhamnogalacturonane, RG1 and rhamnogalacturonane type 2 primary cell wall reported from the grass family Poaceae. This shows presence of cellulose, hemicellulose, pectin approximately 10%, homogalacturonone HGA, rhamnogalacturonone RG1 and RG2. There are three major polysaccharides. The cell wall made up of three major polysaccharides, that is cellulose, hemicellulose and pectins. Cellulose. The cellulose molecules provide tensile strength to the primary cell wall. Each molecule consists of beta 1 4-linked linear chain of at least 500 glucose residues that are covalently linked to one another to form a ribbon-like structure which is stabilized by hydrogen bonds within the chain. It is found in form of microfibrils. These are bundles of about 30 thread-like cellulose molecules 10 to 25 nanometer in diameter, orderly arranged in parallel arrays, forming an extended three-dimensional lattice characteristic of these crystals called micelles. Intermolecular hydrogen bonds between adjacent cellulose molecules cause them to adhere strongly to one another in overlapping parallel arrays, forming a bundle of about cellulose chain, all of which have the same polarity. These highly ordered crystalline aggregates, many micrometers long, are called cellulose microfibrils and they have a very high tensile strength. Hemicellulose. Hemicellulose are highly branched long chain of glucose. There are different types of hemicellulose with a different function, that is Xyloglucans, arabinoxylans, glucuronoxylans, galactomannan. Microfibrils are coated with the fibrous hemicellulose like xyloglucans. They are usually connected by hydrogen bond with a cellulose. Xyloglucan is in turn chemically bonded to another hemicellulose that serve as a cross link between pectin molecules. Xyloglucans. It is one of the type of hemicellulose. Xyloglucans, xylans are a family of structurally diverse plant polysaccharides with a backbone composed of 1,4 linked beta D xylosyl residues, and the functions of the xyloglucans are binds the cellulose microfibrils through non-covalent, that is hydrogen bonding interactions, coat and cross-link adjacent microfibers and serve as load-bearing glycans in primary cell wall. Xyloglucans serve as source of signal molecule. For example, XXFG counteracts auxin-induced cell expansion. Xyloglucan also serve as seed storage carbohydrate. Arabinoxylan, the predominant hemicellulose of grasses. The arabinophyrans attached randomly by 1-alpha 2, 1-alpha 3 linkage to the xylose units throughout the chain. Side chains containing arabinosyls, galactosyl, glucosyluronic acid and 4-O-methyl glucosyluronic acid residues have been identified. Glucuronoxylan. It is a major component of secondary cell walls of dicots. It is present around 15 to 30%. In this, alpha 1,2 linked D-glucuronyl, that is GLCA, 4-O-methyl GLCA, that is MEGLCA, residues attached to carbon every 10 xylene residues. Approximately 70% contain 1O acetyl group at C2 or C3. It contains a distinct glycosyl sequences at the reducing end. It is devoid of ARA units. Galactomannan. Galactomannan are food reserve polysaccharide in endosperm of legume seeds and in endosperm wall and cells legumes. These reserves carbohydrates are used during seed germination, protect the seed from desiccation and are used as a thickeners and stabilizers in the food industry. Galactomannans are beta 1,4 linked mannans substituted by alpha 1,6 linked gal. Pectin is a family of complex carbohydrates found in all plant primary walls that play structural and informational roles in plant cells. Pectins are a heterogeneous group of branched polysaccharide that contain many negatively charged galacturonic acid units. Because of their negative charge, pectins are highly hydrated and associated with cloud of cations. Cellulose and hemicellulose are embedded in cellulose hemicellulose network with pectins. Types of pectins. In plants, different types of pectins have been reported. Some of them are as follows: homogalacturonone HG, xylogalacturonone that is XGA, epylogalacturonone that is AGA, rhamnogalacturonone that is RG1 and rhamnogalacturonone 2 that is RG2. Homogalacturonone Hg is the most abundant pectic polysaccharide, is a homopolymer of alpha 1,4 linked galacturonic acid that may be methyl esterified at carbon 6 and acylated or xylosylated at carbon 3 position. X-ray diffraction studies indicated that homogalacturonone adopts a 4.45 right-handed helix, which is shown in the figure. Functions of pectins. Pectins forms a gel in the presence of divalent cations, that is carbon ions, or in acidic condition in the presence of high solute concentration, for example sucrose. Homogalacturon calcium complex contributes to cell wall strength. In the following picture, we can see how calcium helps in bridging pectin molecules, which gives strength to the cell wall. The figure shows connections among major component of a cell wall, that is glycoprotein, cellulose microfibril, acidic pectin molecules, hemicellulose molecules, neutral pectin molecules, and calcium ions helps to bind the pectin molecules. Next component is protein. In addition to two polysaccharide based networks that are present in all plant primary cell walls, proteins contribute up to about 5% of the wall's dry mass. There is presence of structural and enzyme proteins, approximately 10% of dry weight. Many of these proteins are enzymes, for example peroxidase, cellulase, pectinase, phosphatase, responsible for wall turnover and remodeling, particularly during growth. Hydroprolines, a rare amino acid found in plasma membrane, bound to polysaccharides of the cell wall forming glycoproteins, rich protein. These proteins are thought to strengthen the cell wall and they are produced in greatly increased amount as a local response to attack by pathogens. Plant cell wall proteins. Structural proteins. The major part of the cell wall proteins functions to contribute the cell wall strength, control cell wall assembly, expansion of the cell, hydration, permeability, serve as a possible nucleation site for lignification and as a source of signaling molecule. Other proteins are in small amount including the enzymatic, lipid transfer, signaling and defense proteins. First, structural proteins. Based on the enrichment of specific amino acid and the presence of repeated sequence motifs, they can be classified into two groups, the glycine rich proteins, that is GRPs, and the hydroxyproline-rich glycoproteins, that is HRGPs. Hydroxyproline-rich glycoproteins, that is HRGPs. HRGPs, the major component of structural cell wall protein. They are all glycosylated and contain hydroxyproline, that is HYP. Table shows the protein class and the details of the different proteins. Enzyme proteins. Enzymes like peroxidase, cellulase, pectinase, kinase, phosphatase are associated with wall and are responsible for turnover and remodeling, particularly during cell growth. These proteins are thought to strengthen the cell wall and they are produced in greatly increased amount as a local response to attack by pathogen. The middle lamella. Middle lamella cements together primary cell wall of two adjacent cells. It is mainly pectic in nature, but often becomes lignified in other cells. Lignin, a complex chemical compound polymer, gives rigidity to the cell wall. Middle lamella, a region between cells, is composed of pectin and glues cell together. It is hydrophilic, holds up to 65% of water in primary walls, which is hydrophilic in nature. Figure shows the structure of middle lamella secondary cell wall, where the middle lamella visible as the yellow lines that occurs where adjacent cell wall meets. The red color indicates the presence of lignin in the secondary walls. Figure 2, that is lignin matrix. The pectins can be replaced by lignin, which is impervious to water. Lignin add a tremendous amount of strain to the cell wall and also make them inflexible. This slide shows matrix components of the cell wall. As we know, basic cell wall shows presence of cellulose microfibrils embedded in highly hydrated matrix. Matrix consists of hemicellulose, pectins and small amount of structural proteins. Matrix is highly hydrated gel phase in which cellulose microfibrils are embedded. This figure shows electron micrograph showing Golgi stacks and vesicle containing xyloglucan, which is shown by large arrows, and glycosylated proteins, that is shown by small arrows. These sections taken from sycamore suspension culture cell was labeled with the two types of antibodies conjugated to colloidal gold particles. The large particles are attached to the anti-xyloglucan antibody and the small particles are attached to the antibody that detects glycosylated proteins. To summarize this module, we have learnt about the cell wall. Cell wall is the important component of plant cell. Plant cell is divided into two types, primary cell wall and secondary cell wall. The cell wall compositions are cellulose, hemicellulose and pectins. As we have studied, plant cell wall plays important role in giving tensile strength to the cell wall, and it also regulates the size and shape of the cell. In this way, plant cell wall plays an important role in plant cells. Thank you.